coming to the evolution of sequencing technologies. So we will see how sequencing technology evolved over time. So when we say sequencing technology, you must have heard first generation, second generation, and third generation. Also, during the inauguration, sir mentioned about the third generation, which is ONT, Oxford Nanopore, and PacBio, right? So now let us have an understanding about all three generations of technology. So this first generation of sequencing technology, which was developed by Maxam and Gilbert and Sanger. This is also referred as Sanger sequencing or chain termination method. This was initially developed by Frederick Sanger back, I guess, in 1977. Okay, so far it was a great groundbreaking research back then, and still I would say this sequencing, Sanger sequencing, is still in use when we have to know the sequence, DNA sequence, 100% accuracy. With 100% accuracy, then even in clinical applications we use. But the limitation of this sequencing is it can sequence only up to 1,000 base pairs. And even in 1,000 base pairs, I would say the first and last region of the sequence, you don't see clear bands. I'll explain about the Sanger sequencing in a while in detail, but you would not see that. So roughly you end up having the 700 bases of sequence, but it's compared to other two, compared to at least the second generation, I should mention the Sanger sequencing is highly accurate. Then coming to the Sanger sequencing, the sequencing process, it starts with having the reaction mixture that has DNA template, primer, DNA polymerase, then DNTPs and DDNTPs with fluorochromes. So first, let's understand what is this terminology. So DNA template, when we say DNA template, it is nothing but a DNA sequence, DNA that you want to have, that you want to sequence. Then coming to the second one, primer. The primer is a short and a single stranded DNA which goes and binds to this template. Okay. Then DNA polymerase. DNA polymerase is an enzyme that helps in the chain elongation of the template. But this DNA polymerase, it needs DNTPs, which are nothing but DATP or DCTP, ATGC. Okay. So this DNA polymerase, it incorporates DNTPs during the chain elongation. But what happens? Okay, let's first understand the difference between DNTPs and DDNTPs. Okay, so DNTPs, they have it. It is a deoxyribonuclease, and in a DDNTP, the hydroxyl group at 3' end is not present. So because of that, what happens whenever this DDNTP gets incorporated in the during the sequencing process, the chain gets terminated there itself. So it cannot proceed further. But good thing is this is attached with the fluorochrome. So what happens by the end of this chain reaction, you end up having a bunch of DNA fragments of different lengths. So I would again explain this procedure. So what happens? This DNA polymerase, it incorporates DNTPs to the sequencing process. Whenever it encounters DDNTP instead of DNTP, the chain gets terminated there itself, and we end up having a bunch of DNA fragments of different sizes. So now these DNA fragments are passed through the capillary gel electrophoresis. What happens? These shorter fragments, they run faster, and then the longer fragments, they run slowly. No matter what, so this laser, it detects the fluorochrome attached with the DDNTP and gives us a peak here. So this is how we end up having a sequence in the form of, as a peak, as a chromatogram. So here you see the sequence. So each peak represents one base. For example, here blue represents maybe C or something. Okay. So this is how we get the DNA sequence. It is highly accurate. So the advantage of Sanger sequencing is, first thing is it is highly accurate, and it is very ideal for smaller DNA fragments, for example, individual genes or gene fragments. But the disadvantage is it is very low throughput. It means we can only sequence up to 500 to 1,000 base pairs. So since because of its low throughput, the cost and the time increases significantly for larger genome. So coming to the second generation sequencing. Unlike first generation sequencing, in the second generation sequencing, it is high throughput because of the parallelization of sequencing reactions. So they say the second generation sequencing can sequence up to 500 base pairs, but a typical Illumina platform, it sequences 150 reads, 150 bases in its read. So a read that gets generated from a Illumina platform has 150 bases. So yeah, why the second generation sequencing, or otherwise it is also called short read sequencing, why it brought the revolution? Actually it was initially UK based company Solexa, later acquired by Illumina. It brought a key innovation which is also known as bridge amplification. So what happens in the bridge amplification? It allows the formation of clusters of amplified fragments across the flow cell. We will understand this process in detail in next slides. So I will briefly explain you about the major steps in sequencing. The major steps in sequencing, the first one is sample preparation, followed by library preparation and sequencing. Then after that comes the data analysis. So in sample preparation, this involves the isolation of high quality DNA or RNA from your sample of interest. So the sample of interest can be anything. It can be your patient sample, either a blood sample or saliva, or your plant sample or bacterial genome. It can be anything. So isolation is the first step. You have either DNA or RNA. Then I'm briefly talking about the steps. We will we will understand them in detail in future slides. So the second one is the DNA is fragmented and adapters are ligated to the ends of the fragments. This creates a library. Then this prepared library is loaded onto the sequencing platform. So how it is done? First step is sample preparation and quality check. So as I mentioned, the DNA or RNA is isolated from different types of samples. Then quality checking is done in different way for the DNA and RNA. So why the quality checking is important before proceeding for the sequencing? It is essential because the starting material should be free from the contamination and it should not have a degraded starting material. You know, the quality and quantity also should be good enough for the platform. And then the quality checking is for the DNA. It is done by A280 by A260, and for the RNA it is done by RIN values. So then the fragmentation is done either by enzymatic way or any mechanical way. So now what we have isolated the sample. Let's stick to the patient sample for now. So now we have isolated the DNA sample from the patient blood. Let's assume that. Now after that this sample is isolated, then its quality should be checked using A280 by A260. So it should have an optimal quality, and also the quantity. For example, let's say Illumina. Let's stick to the Illumina also. Illumina also has different platforms like Nextflow, then then different platforms it has, right? So according to the platform, Novasec or so not Nextflow, Nextsec or Novasec or Minisec. So according to that platform, we should have enough concentration of DNA, right? So the concentration and also quality should be checked. So once first check is done, we fragment the DNA either in enzymatic way or mechanical way. Then the second step is adding the adapters. So the adapters, what are the adapters at first? So adapters are short oligonucleotide sequences that are ligated to the DNA fragment. So this is the DNA fragment. The blue color that you see is the DNA fragment, and adapters are attached on both ends of the DNA. See, the green and the pink color are the adapters. So what is the need of adapters? There are three reasons. The first one is we want the DNA fragment to go and bind to the flow cell. Okay, I will explain what is flow cell in a minute. So then, and the second purpose is we can actually club together multiple samples. So, but for that, we should have a unique index sequence, which in other terms, we call them as I5 or I7 indexes. So what happens for each sample, we attach one kind of index, so that, and so since each sample has its own unique index sequence, when we club together, when we pull together multiple samples, or which other way we call them as multiplexing. So that doesn't, and we can proceed for the sequencing at a time, and they do not get mixed with each other. So the adapter has the unique index sequence, which helps us in pulling multiple samples together in the same sequencing. Of course, this will be later demultiplexed before starting the data analysis procedure. Then, okay, let's have a look at the adapter sequence. So at first, it has, this is the DNA insert, as I mentioned earlier, the blue color one. And on the left hand side, the green region is the region complementary to the flow cell oligo. I will show you the flow cell in next slide. So this is the complementary to the flow cell. So what, since this is a complementary region, now this fragment can go and bind to the flow cell. Then it has the index 2, and here it has the index 1. These are nothing but I5 and I7 index. Then followed by it has a primer. So primer is where, this is a primer binding site. This is where the primer can come and bind and then sequence the DNA fragment. Then below here, you can see the flow cell. This is how the flow cell, maybe in zoom it looks like. The flow cell, it contains two types of oligos. Both, what we see here, the both types of oligos are attached. Similar, this way we have millions of oligos attached to the flow cell. So what happens now when we add the DNA to the flow cell? The DNA fragment, it goes and binds to the oligo in the flow cell. It hybridizes. In other words, it is called as hybridization. Hybridization of DNA fragment to the complementary oligo on the flow cell. This happens. So once this DNA fragment is bound to the oligo on the flow cell, the DNA polymerase, it creates a complementary strand against this original fragment. So now we have a complementary strand, as you see here. After that, what happens? The double-stranded DNA, it is denatured, and the original fragment is washed away. So this one is washed away. So now, as you see here, we have this fragment, but this strand, it folds over and binds to the another adapter region. Okay. Now again, what happens after this? Another DNA polymerase, it generates a complementary strand and forms a double-stranded bridge. This way, we have millions of bridge on a flow cell. So after that, this bridge is denatured and it produces multiple single-stranded molecules, as you see here. Okay. So now we have multiple single-stranded molecules. After that, the sequencing by synthesis starts. So what happens? The fluorescence labeled nucleotides, they get incorporated. As you see here, the fluorescence labeled nucleotide, it gets incorporated to this DNA strand by polymerase enzyme. So then whenever this base gets incorporated, it emits a fluorescent signal. For each base, we get a different kind of signal. For example, this is A, T, G, T. I mean, we get different kinds of signal which the software recognizes and gives us a base. So typically, there are two types of sequencing. One is single-end sequencing. The other is paired-end sequencing. What happens in single-end sequencing? So here, the DNA fragment is there. Let's say, assume this is a DNA fragment and it is read only from one end. Okay. But in, as I showed here in this slide, so here, this is the DNA fragment, right? So if it is read only from one side, this is single-end sequencing. But if the same DNA fragment, if it is read from both sides, that is called paired-end sequencing. So in the single-end sequencing, the sequencer, it generates single continuous sequence for each fragment. So you can see multiple fragments, and they should also, we call this as a chromosome walking, if I'm not wrong. So this should get organized this way, so that then only we can have a continuous sequence. Whereas in parent, this sequencing happens from both the ends, and this results in 2 reads per fragment. And this is also called as read pairs. That's why whenever we have a sequence from Illumina or any other platform in paired end, you see R1 and R2. It is a read 1 and read 2. Then comes the last one, the third generation sequencing technology. So in third generation sequencing technology, we have Oxford Nanopore and PacBio. So Oxford Nanopore, unlike the second generation sequencing, here the sequencing happens in real time. So what does that mean? We will understand in a minute. So here for sequencing, we have a biological or synthetic pore that is embedded in the membrane. Okay. So this nanopore is attached to the motor protein. So what this motor protein, it acts as a helicase. So what it does, it unwinds the DNA. Okay. So it unwinds the DNA, and the single stranded molecule, it passes through the pore. Okay. It passes through the nanopore. So whenever a base passes through the nanopore, there is a difference in ion current. So there is some change in ion current which gets captured, and it gives a pattern that is called as squiggle. So this one, for each base, it gives a different kinds of pattern. For example, AT, CG, as you see, different types of drop we see in the sequencer. So that's why it is a real-time sequencing we call. And where this third generation sequencing is applied more is to understand the structural variant, to understand the structural variant, to detect the structural variant. So at first, what is structural variant? So you see different types of variant. I told you what is variant, right? So it is the variant means you have a sequence, a reference genome, but you have a patient DNA which is not like reference genome at particular base. Then that particular base is called a variant. Okay. So if it is a single nucleotide, we call it as a single nucleotide variant. But what is structural variant? When you say inversion or translocation, or so what is inversion? What is translocation? So what happens if instead of a single nucleotide, a part of the a part of the gene or a genome gets mutated. Okay. In case of inversion, what happens? A part gets inverted next to each other, or it can be at different location also. So what happens in the short read sequencing? Since it sequences only for a short read, it cannot actually capture it. It doesn't know where this read is coming from. I mean, even if it is an inversion, maybe it will not align properly because it is a short read, right? So, but that kind of problems can be addressed by third generation sequencing by using long read sequencing. Especially the Oxford nanopore is used for detecting long structural variants. Then epigenetics. As I told, this is a direct sequencing of DNA and RNA. So if a DNA or RNA has the methylation methylated base, for example, 5' methylated base or whatever, so that can be recognized using the nanopore. But it's difficult when it comes to the other platforms. And the mostly we must have heard during the COVID, the nanopore was in it was put in most use, you know. So it does the rapid sequencing of microbial genomes and also any virus genome. So for real-time pathogen detection. Apart from this, if you want to study the full length transcript, okay, then also you can use the nanopore sequencing. So the benefit of nanopore sequencing is the sequencer is portable. For example, it has 3 types of sequencer: MinION, GridION, and PromethION. The MinION, it looks like a pen drive, and GridION is smaller one, a bench like a desktop version, you can say. And PromethION, which is a little bigger version of GridION. Then in the third generation sequencing, we have another one called PacBio sequencing, which is I mean the most accurate sequencing currently in the market, I should say. So here you have the double-stranded DNA, as you see here. So, and of course, before that, let me say this sequencing is called single molecule real-time smart sequencing. Okay. Unlike for Illumina, here for the DNA isolation also, the specific kit from the PacBio only comes. So DNA is isolated because we want the long reads, right? So DNA is also isolated using a PacBio specific kits. Then adapters are attached. These are, you see, as you see, these are hairpin adapters on both ends, right? Hairpin adapters are attached to the double strand DNA. Then a primer and a polymerase. You see the green color is the primer and a polymerase. These are added. So when they added, uh, the sequencing starts. This makes this library. What happens in real time sequencing? The polymerase, it incorporates the nucleotides and forms a consensus, a circular consensus sequence. So it keeps on incorporating the nucleotides and making a circle. And what you take here is a consensus sequence. So a mistake, as they say, a mistake cannot happen twice at the same position. So if you see a difference in the base, different from the reference genome, and if the reads are coming from PacBio, you can blindly trust that it is a variant, because as I said, a mistake cannot happen at the same place, at the same position twice. So, but if it is happening, it must be a variant. If you are not clear, I will explain again. I'll take the questions. So, uh, and the reads that are coming from PacBio, they are called hi-fi reads, and they are highly accurate long read sequencing currently. So, if you look at the difference between ONT and PacBio also, both produces the long read sequences, and it can, in PacBio it can go up to 10 to 20 kb, average is 15 kb, and accuracy in a nanopore is compromised. But that is why it is used mostly for structural variants, but not for the single variant detection. Whereas PacBio is very highly accurate, 99 greater than 99% accuracy we see here. And the cost, nanopore is very affordable, I should say, and PacBio is highly costly. And nanopore is portable, and it does this real-time sequencing. And here, high fidelity, and it gives us the circular consensus sequencing.